Okay, folks, let's get started. What's the word of the day? Happy Friday. Very good. You guys are trainable. Okay, so it is a happy Friday. It's good. So I hope everybody's having a happy Friday. We have a 3-day weekend. 4-day? Well, 4-day from this, that's for sure. I guess we have a 5-day weekend from this because we don't come back until Wednesday. So, wow, awesome. All right. Let's see, as I said, last time I'm behind, and I'm still behind. And this time of the year, I need to sort of rearrange those lectures. But we're in good shape with stuff. I'm not worried about that because I gave an extra day before the exam. All right. What I want to do today is just say one more brief word about membranes. And then we'll turn our attention to the mitochondria, where an understanding of membranes is very, very important because, of course, the mitochondrial membrane is where all the action is. One of the things I didn't talk about last time, I told you something and I didn't really give you anything to think about with it, and I want to do that with you here. We talked about nerve cell transmission, and we talked about how that signal is transmitted. And I said that first we had a sodium gate that opened and the sodium ions came flowing in, and then we had a potassium gate that opened and the potassium ions went flowing out. But there's something for us to think about with this. A sodium ion is a single atom, and a potassium ion is a single atom. And they're not the same size. They're not the same size. Potassium ions are larger. Their atom size, their atomic size, is larger than that of a sodium ion. So how in the world can we envision that we make a little chamber that lets one through but not the other? Okay? You see the problem? So we can't just simply have something that is size excluded. Okay? There has to be other considerations in what lets one but not the other. Okay? Well, it's pretty easy to imagine that for a sodium channel, if the sodium ion is smaller, we actually could use size exclusion because the sodium channel, that little gate that opens up, if it's only big enough to let a sodium go through but not to let a potassium go through, it works. And it does. So for the sodium gates, size exclusion is the way that potassium ions are kept out. And it's very effective at keeping potassium ions out. Maybe 1 in 1,000 to 1 in 10,000 ions make it through as potassium. The rest are all sodiums. For the potassium channel gate, we don't have the same option. We don't have the same option. Because if we make it the right size for the potassium, the sodium is smaller, it's just going to go scooting through. So for the potassium gate, we have to have some other consideration that limits the movement of sodium ions through it. And this is always a little difficult for students to understand, so, and I'm going to go through it a little fast, so if you want to talk more about this, please come see me. But I will tell you what's happening here, and it's really interesting. Okay? What happens is there is an energy exclusion that must be considered with how sodium ions are kept out of the potassium gate. Okay? Now, this exclusion happens not as a result of the size of the ion, but rather the size of the hydrated ion. Okay? So here we are. Here's sodium size, here's potassium size, and we see these various things. Okay? So sodium is smaller than potassium. It turns out that these ions, if you think about it, water has partial charges. Water has a partial charge that is attracted to the positively charged ion. So here's the negatively charged oxygen parts of water that is attracted to that partially charged positive ion. So it makes a sort of a solvation sphere, as it were. That potassium is surrounded by waters. Okay? When the ion makes it to the chamber, when the ion makes it to the gate where it's going to be let through, what has to happen is the waters have to come off first. That takes energy. Okay? Waters have to come off. So it's showing what's called desolvation energy, and that's the energy necessary to strip those waters off before this guy goes through. Excuse me. As the molecule is making its way into and through the chamber, waters come back on. Okay, so we have an energy to remove it, and we have an energy that's released as the waters come back on. Now you'll notice here that the energy that's released as the waters come back on for a potassium is negative, meaning it's favorable. That we see that there's more energy realized by putting them back on than there was in stripping them off in the first place. Now, the biophysical reason for that I'm not going to go into. I'll just briefly tell you that it really has to do with the geometry of the channel itself. The positioning of those waters within that channel is perfectly favorable for an ion the size of potassium. Perfectly favorable. So it's for this reason, because this channel has the exact dimensions that works for a potassium, that this overall process is favorable for a potassium ion. The same is not true for a sodium ion. Okay? When we look at the sodium ion making its way through, what we see is that the desolvation energy, that is the energy necessary to strip the waters off, is here. But because the sodium ion is a different size, the positioning of those waters in the chamber is not as favorable as it is for the potassium, and we don't realize as much energy. Notice we see now a positive delta G energy, meaning that this process is energetically unfavorable. So this is how the energy consideration is used to keep the sodium from passing through the potassium channels. This works pretty well, anywhere from about 1 in 100 to 1 in 1,000 of the sodium ions make it through. Okay? Potassium beats it by quite a ways. Not as much as the other one where we had size exclusion, but it's still a pretty good selection. So the key here is energy, and that energy arises, the energy difference of solvation arises because of the geometry of the chamber and the size of the ion. That probably raised a few questions in your minds. If anybody's still awake. No. Makes sense? You can explain this to me on an exam? Yes, Robert.
Is it ever possible that when water is attached to ions that it can pull it through the membrane through aquaporins?
Is it possible that what? I'm not sure I understood.
That, well, because you said that H2O bonds to the ions, right?
Mm-hmm.
Would it be possible that the aquaporins that allow water?
Oh, so you're talking about the aquapores that allow water through. No, those are going to be specific for water. So if you start putting something, yet another atom on there, it's going to be, that's enough to exclude it easily. So no, that's not the way those would come in. Yes, Danielle.
So just to clarify, before they pass through this channel, they have waters attached to them. The waters have to come off.
Yep.
They go through the channel, and while they're in the channel,
Waters come on.
get back.
Right.
That is back on.
That's right. Yep. So the geometry of that channel is critical to this process. Yes, Shannon.
What are the non-water molecules in the channel on the picture?
The non-water molecules in the channel on the picture. So those are side chains of the amino acids that are part of that protein. Right. So again, the positioning of everything in this channel is very critical for this overall geometry, and the side chains of the amino acids in that protein help to orient that. Good question. Excuse me. Yes, sir.
What takes the water off?
Yeah, what takes the water off? It's actually a very complicated process, as you could imagine. But the entry into the chamber, the ions are literally pulled into the chamber, and it's that movement through the chamber that strips them out.
Is there just water diffused all through the chamber that randomly reattaches as it goes through?
Right. So water has to have access to the chamber in order for this process to happen. And so that water can come in from either direction. So it can come in from the backside once you've let one through. It can come in that front direction as well.
So the entrance is definitely a different shape than the rest of the channel. For the entrance,
Is the entrance a different shape than the rest of the channel? Well, I don't want you to think of this as like a perfect tube, because remember we're talking about a protein. And at the molecular level, we've got various things sticking out in various places. So no, I would not say that. Every part of the channel is different in that sense. Okay? Yes, question up here.
What's the driving force pushing this through? Like, I understand the reaction is favorable, but what gets it in there to start with?
Okay, so what's getting us in this to start with? I'll ask the class that question. What gets us? Why does it want to go in there? Concentration. Because remember, we've got a concentration gradient. We pumped all that sodium out, and the sodium pumped all the sodium out and all the potassium in. So once we open that channel, then that concentration gradient is going to push it. Yes, sir?
Where did that extra energy come from the potassium?
Yeah.
Free energy.
Yeah, again, as I said, this entry of this process is very complicated, so I'm not going to go through it. It's not an ATP energy. It actually is an energy arising from the movement of that to and through that. Okay? But ultimately, that energy is coming from a concentration gradient. That's the ultimate answer to your question. Okay. Oh, one more question.
Does the water,
This is a good question. You guys have been asking a lot of good questions there.
does the water stick on the ion when it goes through, or is it just the ion that goes through, or does it go through with the water?
It goes through. No, it gets stripped. That's the desolvation.
Or in the channel? Or do you attach
And
it?
in the channel, it's reattaching.
So when the ion flows through the channel into the cell, does the water stay attached to it?
Yes, yes. It's resolvating, so it's regaining ions. Yeah, yeah. Okay. Well, that's the last of our membranes. So at least last of the detail on our membranes. I want to turn our attention now to talking about the mitochondria. And all these considerations we had about pumps and things like that should obviously, hopefully, now start to make some sense. I've previously talked about the mitochondrial structure. I'll just sort of show you schematically there that process. We had the outer membrane shown here in yellow. We had the inner membrane shown in blue, and we see the inner membrane has inner foldings called cristae. And these inner foldings give a lot of surface area to that inner membrane. The more surface area the inner membrane we have, the more energy that membrane can actually help to produce. And we'll see how that goes. Last, we have the matrix, which is the sort of liquid cytoplasmic-like chamber in the middle of everything. Okay? The inner mitochondrial membrane is impermeable to virtually everything except for oxygen, water, carbon dioxide, carbon monoxide, just like a regular membrane. Okay? Those four things can readily move across the inner mitochondrial membrane. Things as small as a single proton cannot. The inner mitochondrial membrane is impermeable to protons. Yes, ma'am? Yeah, the four things that don't make it across?
Yeah.
Yeah, so the four things that don't make it across. Water, oxygen, carbon monoxide, carbon dioxide. They all readily move across the mitochondrial membrane. Protons do not. And most everything else does not.
Is water moving through pores, or is it actually moving through the lipid?
Water can move through lipid bilayers quite easily. Yep. It's surprising, but it can.
I forget.
Okay, now, so the key feature in understanding the mitochondrion is the fact that the inner membrane is very impermeable to most things. And protons, as we'll see, are a major factor here. The outer membrane is permeable to most things. And people say, well, why do we even worry about it? Well, it's sort of, I like to think of the outer membrane as like a wet blanket over everything. A wet blanket holds things in, but a lot of things like water and so forth will readily go through it. Okay? So a wet blanket sits on you, and it actually helps to hold some heat or other things in, but it really isn't very good at keeping you dry or another way. So the outer membrane of the mitochondrion is sort of like that. It's permeable to a lot of things. In fact, it's permeable to most things up to about 50,000 in molecular weight. So it lets a lot of stuff through. But it will have the effect of sort of holding things relatively close to the mitochondrion just because it's laying on it like a wet blanket. Okay. What happens to the mitochondria? We have two major considerations for what's happening. The mitochondria, of course, is what we refer to as the power plant of the cell. It's the place where most ATP is made. And the action is all in the inner mitochondrial membrane. That's what you see there in gray. Excuse me. Okay? Two processes to consider that happen there. They are independent, but also interdependent. All right? The first process is electron transport. That is schematically shown by this yellow thing here. The second part of that is called oxidative phosphorylation, and that's shown by the purple thing here. Now, as we'll see, especially the electron transport, there's multiple steps in that. It's not just one step, and we'll have to take that into consideration as we go through it. Okay. Now, Electron transport. So I think most of you probably have had electron transport in basic biology. I'm going to go through it in a little bit more detail than you've seen in basic biology, but I'll just start at the beginning for anybody who's not seen electron transport before. Okay? So electron transport is literally an electrical circuit in the inner membrane wall of the mitochondria. It's literally an electric circuit. An electric circuit allows electrons to move through it. They move from a high potential to a lower potential. Okay? And that's exactly what is happening out here in the mitochondrial inner membrane. Okay? There are a series of what are called electron carriers that facilitate the movement of electrons. Now let me caution you as I get going through, before I even say anything, don't confuse electrons and protons. I will do it in my lecture. If I do, please tell me so I don't confuse people. But electrons and protons are doing very different things in this process. All right. For the moment, we're going to focus completely on electrons. Why is electron transport important? Well, electrons, remember we have NADH. Cells have made NADH because there's been a lot of oxidation that's gone on. Those electrons from the oxidation were transferred to NAD to make NADH. And we've got to get rid of the electrons that are in that NADH so we can regenerate NAD. If we don't, the cell is hosed. So, electron transport serves to recycle NADH back to NAD. The process happens as follows. NADH, which happens to be in the matrix of the mitochondria, transfers a pair of electrons to the first electron carrier. Okay? Electrons in the process start moving in pairs. They start in pairs. So they're coming off of NADH as a pair. And those electrons are donated to this NADH-Q oxidoreductase. I don't like that name. So let's just call it complex 1. Okay? So we transfer electrons from NADH to complex 1. You guys are all ready to name it. I'm going to give you easier names than Virginia. I think you're going to like my names better than your names. How about that? We can take a vote. If you guys want to give it other names after you've heard my names, then we can decide that. Is that fair? Yes, ma'am.
Which one is complex 1?
The very first one, NADH-Q oxidoreductase. Okay? And by the way, the names that I'm giving these are not names that I made up. They're actually names that are used by most books. So you'll see what I'm going to tell you here in most places. All right. NADH, I can't even say it. Complex 1 has now accepted a pair of electrons. Complex 1 is a big honking complex. Many proteins in this complex. It's sitting there. It is an integral membrane complex because it's just sitting there in the mitochondrial inner membrane. It's not very mobile. Complex 1 transfers its electrons to this little guy here called Q, more commonly called coenzyme Q, or CoQ, C-O-Q. CoQ is a small molecule. It is very mobile. It can move in the mitochondrial membrane very readily, and that's very useful, as we shall see. Coenzyme Q accepts electrons in pairs. So in other words, it's taking a pair of electrons from NADH, but it only passes them off one at a time. Coenzyme Q acts like what I call a traffic cop. A traffic cop decides how many cars make it through and directs everybody how to get somewhere. Coenzyme Q says, okay, bring me the 2, and then I'm only going to let 1 through at a time. And that's important because the other complexes only handle electrons 1 at a time. So they get sort of sorted out by coenzyme Q. Coenzyme Q transfers its electrons to Q, cytochrome C oxidoreductase, and that's a big mouthful of a name. Let's call that complex 3. But wait, where's complex 2, Kevin? Right? There's complex 2 right there. We'll see how that plays in just a moment. Shirley? Shirley, you're kidding, or Shirley the name? Yeah. All right. All right. Complex 1, complex 2, complex 3. I'll let you guys guess what this one's going to be called down here. Okay. Complex 3 transfers its electron, 1 at a time, to a protein called cytochrome C, or Cyt C. Either is acceptable. Cyt C passes its electron off to cytochrome C oxidase, more commonly called complex 4. Now, we can have Virginia, we can have Shirley, we can have Jennifer, but I think complex 1, 3, and 4 are probably easier to remember. But I'm open to suggestions. How many would like to have people names versus... No. Okay. All right. He said it. All right, so he's agreed. All right. So complex 1, 2, 3, 4. And as I said, you'll see this in most textbooks. Now, last. Oh, yes.
Which one was 2?
2 is this guy right here.
Okay.
I'll say more about it in just a minute. Okay? Complex 4 passes its electrons off to the terminal electron acceptor. This is the thing that accepts electrons in their final destination, the oxygen molecule. When oxygen accepts electrons, it also accepts protons and makes water. Now, last term we talked about why, when we run out of oxygen, we no longer can convert NADH to NAD. I think you can see that if we have no place to put the electrons, this process is going to back up and get gummed up. When this process gets gummed up, we can't convert NADH to NAD. That's a very, very important consideration. Question over here.
Complex 4 transfers 1 electron to oxygen?
1 electron at a time. It will ultimately transfer 4, but it transfers them 1 at a time. And I'll show you that in a bit. Yes, sir?
Does this all happen within the membrane, or are there any extra members made?
Everything happening here, once it has left the NADH, is occurring in the membrane. Everything. I'm sorry? The inner mitochondrial membrane. That's correct. The inner mitochondrial membrane. Yeah. All right. What did I tell you about complex 2? Where does complex 2 fit in this scheme? Complex 2 accepts electrons from FADH2. So electrons coming from excuse me, from FADH2 come in this direction through succinate Q reductase, which we call complex 2. And if you remember succinate Q reductase, that was the enzyme that was in the citric acid cycle. We called it succinate dehydrogenase at that point, but it's the same thing. Okay? This is the same as the citric acid cycle enzyme. So complex 2 is the citric acid cycle enzyme. It accepts electrons from FADH2, transfers them off in a pair to Q, and Q passes them off 1 at a time. We have the same thing going on. So electrons coming in from NADH come in from in a different place than electrons coming in from FADH2. Shannon.
Are we going to go over how exactly the electrons are coming to these compounds and how they're going to be transferred?
I will show you some detail on a couple of the processes, but I will not go through all of them. And the reason I won't is because in this guy here, there are dozens of proteins, and even through that complex, the individual movements through there aren't completely understood. Yes, Danielle.
So two electrons come from NADH,
Yep.
from FADH2,
Yep.
and those comprise the 4 that Cytochrome C eventually passes on to those 2.
Doesn't it could be you could have none of this and everything comes here. It doesn't matter. It doesn't matter. Yeah. So the source of the electrons doesn't matter. Yes.
Why can't Q carry the electrons all the way from complex 1 to complex 4?
Why can't Q do this?
Yeah.
As we'll see, it's not in the cell's interest to do that. Part of it is that because these complexes, this guy, this guy, this guy, they're all multi-protein complexes. They're not very mobile. So these little carriers, Q is very mobile, cytochrome C is very mobile, and so they act as little shuttles between these, and it's much easier to do that. But there's a more practical reason why that doesn't happen as well. Yes, sir.
So FADH to complex 2 and complex 2 to Q, that's 2 electrons at a time?
FADH2, you're talking about here? Yes. 2 electrons at a time. So everything up to coenzyme Q is always done in pairs. And everything after it is done 1 at a time. Okay, so there's our traffic cop. Now, that is what happens with the electrons. This schematically shows you the same process and adds a couple more elements to it. Here's complex 1. Here's NADH. It's been produced. Remember, this is happening in the matrix, so this has been produced in the citric acid cycle. It could have been produced by fatty acid oxidation also. We'll talk about that later. But NADH has been produced in the matrix. It donates its electrons to complex 1. All right, this is actually bypassing 2. It should sort of going around, but it's not going through complex 2. But it's going to coenzyme Q, to 3, to cytochrome C, which is not shown on here, and then finally to complex 4. So cytochrome C is missing off of this figure, amazingly enough. Now, look what also is happening. As electrons are moving through complex 1, we see protons being kicked out of the mitochondrial matrix. So complex 1 is a proton pump. And the energy for that proton pump is the movement of electrons through it. So just like electrons move through the filament of a light bulb and give off light, electrons are passing through complex 1, and what they're producing is a proton gradient. That's pretty amazing. That is pretty darn amazing. All right? Here is a biological circuit. This circuit is taking energy from electrical potential and causing something to get moved. All right? It's a little electric motor. That's really remarkable. And this little electric motor is comprised of proteins. That's cool. That's very cool. Okay. You'll notice that there are 3 places where protons are ejected from the matrix out into the cytoplasm. So the cytoplasm is up here. It says intermembrane space, but for our purposes, that's essentially the cytoplasm. The intermembrane space, you recall, is the space between the wet blanket and the inner membrane. Okay? It's kicking them out here. Complex 1, complex 3, complex 4. There are protons kicked out at each one. There are people who spend their whole lives figuring out how many protons are kicked out at each one, and people argue about this, and it really doesn't matter. Protons are kicked out. Okay? Suffice it to say, become like the old shit happens bumper stickers, right? You say, protons get pumped. Okay, get over it. All right. All right. So protons are pumped. The important thing is that everybody agrees on this. There is a proton gradient that is created as a result of this. The gradient means more protons outside the mitochondrion than inside the mitochondrion. Okay? Something else you notice here. We pump more protons if we take electrons from NADH than if we take electrons from FADH2. If electrons start with FADH2, we only get 2 places where protons are pumped. If we start with complex 1 with NADH, 3 places where they are pumped. Okay? It turns out that if you start proton pumping at complex 1, you generate enough energy for approximately, ultimately, 3 ATPs. And I said approximately, ultimately. You don't see any ATPs here. There are no ATPs made in electron transport. ATPs get made in oxidative phosphorylation. This is doing what I call charging the battery. The battery is getting charged by the pumping of the protons out. We'll discharge that battery and make ATP in oxidative phosphorylation. If electrons start here, and they only go through complex 3 and 4, it turns out there's only enough energy produced for about 2 ATPs per FADH2. Only about 2 ATPs per FADH2. So we can see that the more NADHs we have, the more the more ATP we're going to produce per pair of electrons compared to FADH2. Okay, does that make sense? Everybody with? And these are approximate numbers. Some books will say 2.5 and 1.5. Again, for our purposes, it's not really important. We're going to use approximately 3 and approximately 2 for each one. Okay. That's what's happening in the overall process. Blah, 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 blah. All right. Now, one of the ways people have studied electron transport is through the use of electron transport inhibitors. Electron transport inhibitors are laboratory research tools. They're also poisons. Okay? Imagine if you gum up electron transport, what's going to happen in the cell? Well, now we can start to see what happens. Let's think, first of all, we lose oxygen. What happens if we lose oxygen? Well, we lose oxygen. We're not going to be able to have electron transport, which means, therefore, that we're going to have NADH concentrations go up, What happens when NADH concentrations go up? Citric acid cycle is going to stop, and that's one of our major sources of energy. Of course, ultimately out in the cytoplasm, we're not going to have any glycolysis. We're going to have to go through fermentation, right? Okay, so we can see that stopping electron transport is going to be problematic. Well, there are 3 places in electron transport where we can stop the movement of electrons. The first of these we talk about is blocking the movement of electrons through complex 1. This movement is actually blocked by the compounds rotenone and amytal. Rotenone is a biological insecticide. It's actually produced by plants, and it kills insects. So you can actually go buy, if you go to a garden store, you can buy rotenone. Rotenone is considered completely organic because it is plant produced. And you can spray it on plants and kill insects. It's killing insects by knocking out their electron transport system. It's not really killing people, although I probably wouldn't recommend eating too much of it. Why doesn't it kill people? Well, think about this. If we knock out the movement of electrons from NADH, we still have another place where electrons can come in. Where? FADH2. Okay? So, ideally, we're not going to go around eating rotenone for breakfast, but hopefully we're not going to have this problem. So we can bypass this block by coming in through FADH2. Stephanie?
Do insects not produce FADH2?
They do. They're just more susceptible to this than we are. And we'll actually see an example of that in a little bit. By what? Well, they have more immediate energy needs because, as you imagine, flies, for example, you try to swat them, they fly away pretty fast. So they've got pretty quick energy needs, and their muscles work different than ours do. But suffice to say, that's a good way to kill insects, and it's not a pretty terrible thing to put onto plants, for example. All right. A second place where we can block the movement of electrons is by blocking electrons going from complex 3 to cytochrome C. That is blocked by antimycin A. And while this one is also fairly severe, it turns out that there are other ways of getting electrons in here, but I would not want to have any of this. Okay? Because these other ways coming in, they actually involve vitamin C, are pretty tenuous. All right? If I were to block everything right here, and I said to you, where are the electrons, would you say, where would the electrons be in this scheme? Which things would have electrons, which things would not have electrons? Everything up to there will have electrons. Everything down there from there is not going to have electrons because the water has flowed out, right? Okay. The third place where we block electron transport is by these guys right here. Cyanide. Now you know why cyanide is poisonous. Azide. Azide is very poisonous. Carbon monoxide. Our friend carbon monoxide, which kills us by binding to hemoglobin and blocking oxygen, also kills us by blocking complex 4. Okay? Carbon monoxide is doubly poisonous to us. All right. Now, nothing gets around these guys. Nothing gets around these guys. Okay? You got some real problems if you've got much of any of these in your system, because when that happens, your electron transport system is shut down and you are hosed. Okay. As I said, in electron transport, electrons are moving ultimately to oxygen and making water. If electron transport is not occurring, water is not being formed. Okay. Well, let's take a look at a couple mechanisms. The mechanisms I want to show you involve a couple of things. One is, not my cough, one of the things is coenzyme Q. What you see here is a part of the structure of coenzyme Q. Coenzyme Q is a molecule that's embedded in the mitochondrial inner membrane, and it has a long nonpolar tail shown here in blue. That's the R group right there and there and there. And that long nonpolar tail sticks into the mitochondrial inner membrane. Okay? That's how it's held there, is that long tail. We could think of this as being almost like an anchored protein. It's not a protein, obviously, but it's anchored because it's got a tail that's sticking into that membrane, into the inner mitochondrial membrane. It turns out that coenzyme Q can accept electrons in pairs. It can have an intermediate state where it shuffles those electrons down. It's because of this intermediate state that coenzyme Q can handle one electron at a time. We're not going to worry about structures, all right? And I'm only showing you this figure just to remind you that there are some different states that coenzyme Q can have that can accommodate either 2 electrons or 1 electron. Okay? So it's because of this ability that coenzyme Q functions as a traffic cop for the electrons moving through. Okay? We don't need to go through that. The flavins are involved in electron transport. They're part of the protein complexes. I won't talk about those. Other things that are involved in electron transport that are part of the protein complexes, and when I'm talking about the protein complex, I'm talking about complex 1, 3, and 4. Okay? These complexes have various ways of handling electrons. Complex 1 works mostly with flavins. Okay? Mostly with flavins. Complex 1 has a few proteins involved that have what are called iron-sulfur centers. And iron, you recall, is capable of existing in a +3 and a +2 state. The difference between those two states is 1 electron. And so iron-sulfur proteins participate in the movement of those electrons by the movement of electrons through the iron. You can see these can have some pretty hairy structures. They can be quite complicated. But suffice it to say that each one of these is handling 1 electron at a time, no matter how complicated it looks. Other proteins that are involved in that electron transport system start showing up in complex 3, and they're called cytochromes. Okay? They're called cytochromes, and I don't have a figure here for specific cytochromes for you, unfortunately. Cytochromes are complexes that look a little bit like hemoglobin, and they have iron in them. So they look a little bit like a heme group. They have iron in them, and that iron in them is, again, helping to facilitate the movement of electrons through the complex. Okay. This figure doesn't really tell us too much, and I'm just going to show it to you. There's really nothing on here I expect you to pull out other than the fact that NADH drops its electrons off at complex 1. This is a flavin that's grabbing those electrons. Here's some iron-sulfur proteins that are passing those electrons. And here, and it looks like it's part of complex 1. It's not. It is a sort of a docking chamber for coenzyme Q, grabs the electrons and goes on out. All right? So the electrons are coming through complex 1 from NADH and getting passed to coenzyme Q. When they become reduced, that is when the coenzyme Q has accepted electrons, it becomes what we call here a QH2. QH2 means it's now carrying 2 electrons. From a mechanistic point of view, the important one that we will be concerned with is what happens in complex 3. This shows a schematic structure of complex 3. As you see, these complexes are fairly large. And complex 3 transfers its electrons in something called the Q cycle. Now, the Q cycle always looks and seems like it's a lot more complicated than it is. So I'm going to try to explain it in simple terms for you, and I will take questions if you have questions about how this works. We start on the left with something called the Q pool. All this is a bunch of reduced coenzyme Q and some oxidized coenzyme Q. So meaning some of them have electrons, some of them don't have electrons. That's what the pool is. Since that's the only 2 possibilities, that encompasses all of the coenzyme Q in the membrane. Some have electrons and some don't. Everybody with me? We're going to focus first on the ones that have electrons. These come into the complex 3 as QH2. The complex 3 has been sitting there waiting for these electrons to come in, and the complex 3 has had 2 other things inside of it. One is a coenzyme Q that doesn't have any electrons, and second, it's got a molecule of cytochrome C. That is the protein that does that shuttle that we talked about earlier. Now, these are not physically attached to coenzyme to complex 3. They're just in little chambers within the complex. Okay? Coenzyme Q, with its electrons, comes in. It sends one of those electrons to cytochrome C, because cytochrome C can only handle electrons one at a time. The other electron gets passed to this empty coenzyme Q that has no electrons, and now it becomes a coenzyme Q with one electron. Everybody with me? So the electrons have come in, they've split. One's gone to cytochrome C, one's gone to the other coenzyme Q. In the process of this transfer, protons are being pumped. Protons are moving from the inner mitochondrial matrix to the intramembrane space, or cytoplasm. I'll take either one. Okay? So we see protons moving as a result of this. Cytochrome C, once it's gotten its electron, it goes flying off. It disappears. They don't show that on here. It would be nice if they did that. It goes flying away. Okay? So at this point, we have something that should be shown right here, a complex 3 that has this, and it's getting ready to accept a new cytochrome C that has no electrons in it. We're going to rely on a second cytochrome C to come in that has no electrons in it. Okay? So they missed showing you that step. Now we're going to the last step. Here's cytochrome C. The Q pool allows another coenzyme Q to come in. It's got 2 electrons. And the same thing happens that happened over here. One electron goes to cytochrome C. The other electron goes to this coenzyme Q that already had 1 electron. What did we just make? Okay, we made a reduced coenzyme Q, which now gets kicked out back to the Q pool. Okay? So the net result of this is we brought in electrons from coenzyme Q, and we created 2 reduced cytochrome Cs. Yes?
So the first part, why does an electron get transferred to the regular one laying around? Wouldn't it just...
I'm sorry, say it again.
So in the first part, why did an electron get... why did it do that? Did it just keep it for itself to make itself into a radical?
Well, I don't have an answer for that, but the answer is partly rooted in the fact that there are 2 chambers. So if this guy stays here as a radical, it can't allow another one to come in. And I see what you're saying. Well, why doesn't this just go on out and then another one come in and let its thing go there? That's not the way that it evolved. I don't have the answer to that. Okay. But suffice it to say that because of the nature of the 2 chambers, it's very efficient for just kicking this thing out, putting the electron down here, and then going back through the cycle up there. Okay? Okay. Does this make sense? Shannon?
The proton.
What's that?
The protons.
Yep. Protons are being pumped again. So they get pumped in 2 places in the cycle.
So the QH2, where do those protons come from?
So, okay, so that's a good question. So I frequently get asked this question. Where do these protons come from? Why do I have these protons going out here, and where do these protons come out? Where do the protons come from on coenzyme Q? I've added 2 electrons, but now I'm showing it's got 2 protons too. Where did those protons come from? Aqueous solution. There's always going to be protons in an aqueous solution. At pH 7, there's 10 to the minus 7th molar protons. So there's always enough protons there to protonate whatever we need there. So that's where the protons came from, is they came out of the solution onto there. Okay? You don't look like you're satisfied with that answer.
So is the mitochondria less acidic than the other stuff?
Okay, so that's a good question. So have we decreased the acidity of the mitochondria as a result of doing this? What do you guys think? Yes, we have. We have decreased the acidity because we've kicked protons out. Absolutely. So the mitochondrial matrix will almost always be less acidic than the stuff around it. Good question. Yes, sir?
So when NADH drops its electrons, becomes NAD, what happens to that proton?
Well, see, the tendency here is to say, well, this proton goes here and this proton goes here. But remember, these are all just dumped into the solution. So we're not tracking individual protons in this process. That's right. Yeah, we've got billions and zillions of these protons that are around that can do stuff. Yes, sir?
So if the acidity is low in the air membrane space, what prevents the protons from going into the cytoplasm?
Okay, so if the acidity is low in the intermembrane space, what stops the protons from going to the cytoplasm? If nothing else happens, they will make it to the cytoplasm. And if they make it to the cytoplasm, very many of them, the cell will have a real hard time. And I'll talk about that later. But that is possible to have happen. You betcha. Okay? All right, let's see what I've got here in terms of time. What do you guys say we do a little song to finish off the week? All right, now, I have a challenge for you here. You've heard me coughing, all right? And you've also heard me singing. Two things that are really bad, okay? If I sing, I'm going to cough even more. So I've got a song that we can have this side of the room compete with this side of the room on. I will give you 1/8 of a point of extra credit if you do this, okay? All right. All right, so I'm going to set it. What I'll do is I'll set it up. So we're going to start the first stanza over here, second stanza over here. How many people know Monty Python's I'm a Lumberjack? All right, that's what we're going to sing. Everybody ready? It's called I'm a Little Mitochondrian. Okay? This side over here. Oh, I'm a little mitochondrian. Gives you energy. Come on, folks. I use my proton gradient to make the ATPs. Over here. He's a little mitochondria. He gives us energy. He uses proton gradients to make some ATPs. Over here. Electrons flow through complex 2 to traffic CoQ. Whenever they arrive, they're in an FADH2. Oh! Complex 2 to traffic CoQ. Whenever they arrive, they're in an FADH2. All right. It's tightly coupled is my state unless I get a hole created in my membrane by some dinitrophenol.
Yes, tightly coupled in his state unless he gets a hole created in his membrane by some dinitrophenol.
All right. Proton known at
cyanide, stop my electron flow, and all the calculation of the P2O ratio.
All right, over here. Last.
Electron flow and halt the calculation of his P2O ratio.
Standing ovation, yes. The winners, I have to admit, this side got it. You guys will each get 1/8 of a point of extra credit. So, and don't ask me how I'll determine that. Now, I think you'd have to admit, that's way better than me dressing up in women's clothing. Yeah. All right. Yes, sir.
So I have a question about the neuron signal transduction.
Yeah, come on up.
All right.
I hate this.
Okay. So the key to the traveling is the fact that the neuron, the nerve cell, has many of those gates all the way along it. So we've only talked about the one at the very end.
Right.
All right. So that one at the very end lets in some